Hi everyone. Today we're going to be learning about gene cloning, and so we're going to be taking everything we've learned about DNA and applying it to manipulate genes to do things that we want. So biotechnology is just a term to use that describes when we manipulate organisms or their components to make useful products. This is really general, and this could be applied to even when we selectively breed organisms just so we can get the desired traits. But nowadays, we've taken that biotechnology to a new level by doing DNA technology. And this is when we take modern lab techniques and we use that to study and manipulate genetic material. So, this is specifically looking at DNA. One subset of DNA technology is genetic engineering. And this is when we directly manipulate the genes for practical purposes like creating proteins that we want, doing different things like that. So, to do genetic engineering, we need two pretty important things. One of them is recombinant DNA. Recombinant DNA is kind of what it sounds like. It's just when you combine pieces of DNA from two different sources and you combine them in a test tube, which is called in vitro, and you combine them to form one DNA molecule. So recombinant DNA, two molecules becoming one. And then we're also going to use plasmids. And if you just remember back when we learned about bacteria DNA, plasmids are just small circular chunks of DNA that replicate independently from the main source of genetic material inside a bacteria. And so they're very convenient because they replicate independently, we can use them to create proteins for ourselves. Oh, this picture right here of a fish is an example of recombinant DNA where injected into the fish's DNA were like jellyfish DNA, where that allows the fish to glow in the dark. And then right here is a picture of what the plasmids look like. And so one example of doing all of this is gene cloning is when we produce a lot of copies of a gene. So example of that would be like medicines, like insulin. We can use plasmids and recombinant DNA to do gene cloning and then create a lot of these proteins that we need for medicine. Okay, so the question is, how do we actually do this genetic engineering to produce a mass quantity of some protein X that we want? So how does this happen? The first step is we need to isolate the bacterial plasmid as a vector, which just means that we want to choose whichever plasmid we want to be the gene carrier. And then we also have to isolate the desired gene X that is going to eventually code for the protein that we want. And so here we can see that we are going to just choose some plasmid that we need. And then we're also going to isolate the DNA that we need, and we want to take out the specific gene of interest. After we take these two parts, these are the two different sources of DNA that we're going to combine together. We're going to cut the plasmid at a very specific location, and we're also going to cut out gene X so that we have that fragment isolated. So going back to this picture, after we've taken out this plasmid, we're going to make a cut just in one very specific location. And we're also going to cut out from this isolated DNA the specific gene that we want. And the reason why we're cutting these is obviously because we're going to put them together. Right, so the next step is to take this targeted gene X fragment and insert it into the cut of the plasmid DNA. So when they're put together, the ends of the fragments are going to complementary base pair with each other so that it's inserted in the correct direction. And then afterwards, the enzyme DNA ligase, which we learned about before for DNA replication. Remember, DNA ligase can form covalent bonds, and it will merge these two different segments of DNA together. So right here, steps 5 and 6, we've inserted this gene X into our plasmid, and then you see that the ends kind of complementary base pair with each other so that it fits correctly. And afterwards, DNA ligase makes it a seamless, continuous strand of DNA. And then lastly, now that we have our recombinant DNA plasmid, we're going to mix it into the bacteria so that the bacteria can absorb it into itself. And remember that process of bacteria taking DNA or genetic material from the environment is called transformation. So we're going to transform these bacterial cells and then allow these bacterial cells to reproduce a lot. And so now we have like millions and millions of bacteria that have our desired recombinant plasmid. And then that bacteria will eventually produce the proteins that we want. So we can either do this so that we can have just a lot of copies of just the gene X, or we could do this to get a lot of copies of protein X. So here we take this plasmid, recombinant plasmid, we put it into the bacteria cells, and the bacteria cells will reproduce, and we get a lot of what we want. We have a lot of these genes, and we also can which leads to a lot of the proteins. And so the question is like, well, what are the applications of this? So we might not even want the proteins itself, but we might want to just take the gene. So now we have millions and millions of copies of this gene X. We can insert it into certain things. For example, like we talked about the fish, we can insert that into the fish, and now we can have glow-in-the-dark fish. We can also take certain kinds of genes that are like bacterial resistant and inject them into like plants so that these plants aren't susceptible to diseases, which increases the crop production and makes prices of crops cheaper for consumers. You can also inject genes that will make certain fruits enhance the flavor of it and make it actually taste like a different fruit. So, I mean, this picture is obviously photoshopped, but the idea is like you could take some of the taste of a kiwi and put it into the taste of an orange, and so now you have this hybrid fruit. So this is for if you want to just use the genes, but you can actually, like we said, take that gene, these many copies of the genes to produce many copies of these proteins, and we've talked about how that can lead to a lot of different kinds of medicines, like heart medicine, insulin. There's just a plethora of different applications of that. You can also use it for proteins to make like your genes look faded, and so it has industrial applications, but it also has medicinal applications as well. Okay, so now you might be thinking, how did we actually cut the plasmids in DNA? How did we do that? We used something called restriction enzymes. Restriction enzymes are very specific kinds of enzymes that recognize a specific sequence of DNA and will cut that DNA sequence as soon as it sees it. And so it cuts at these specific places called restriction sites. And then when they do that, they create these fragments called restriction fragments. So here you can see an example where there's a specific restriction enzyme that might just cut like right along here. Notice that it doesn't necessarily do a blunt cut where it'll cut the two strands equally. It might leave actually something called sticky ends, which means that like one of the strands of the DNA sticks out more than the other one. And this is actually really great. These sticky ends are what we're going to use to help base pair when we want to combine things together. So these sticky ends here, created by the restriction enzymes in the fragments, you see how they're complementary to each other. And so when you cut like our plasmids and our separate DNA fragments with the same restriction enzyme, they will have the same kinds of sticky ends, which makes it really convenient to piece them together, and they'll naturally base pair together like we talked about before. Okay, so another application of all of this is we might want to create a genomic library. A genomic library is when we take an organism's whole set of DNA, called their genome, and we cut it with DNA fragments. And then we put those fragments into different plasmids. And so now we have like this easy way to analyze the organism's DNA. So we pretty much just chopped up all of the organism's DNA and separated them. And the reason why we might do this is because it becomes very easy to analyze which specific genes lead to what kind of proteins. So a lot of times you don't necessarily know. And so you can do this, and then you have this array, this like library, and you can check each individual segment of DNA and see what it does. Okay, so let's say we want to do this process of recombinant DNA to mass produce a eukaryotic protein. Can you think of any problems that we might run into if we want to do this whole process to create a eukaryotic protein? Well, something you might be thinking is that if we just take the DNA and we cut out the segment we want, there's still a mix of introns and exons in there. And so if we take that segment of DNA and we insert it into our plasmid, we're not actually going to be producing the protein we want because there's too many introns in there. So, what do you think the solution might be? Well, what if we were able to use mRNA, the mRNA that has already had all of the introns removed, and then kind of go backwards to produce the DNA with only exons in it. And that's exactly what we're going to do. So this is a process where we allow normal transcription to happen. And remember, when transcription happens in a eukaryotic cell, we have just a mix of exons and introns. And so we allow the normal process to happen where those are RNA splicing and all those exons are isolated together so that we have just what we need to produce our protein. And then we're going to take this and isolate these specific mRNAs. And when we take these and cultivate them, now we have our mRNAs and we can actually work backwards. And we're going to use a specific enzyme called reverse transcriptase. It does exactly what it sounds like, where it reverses transcription. And you take your mRNA and now you produce a single strand of DNA that's complementary to the mRNA. After we do that, now we have this weird mix of mRNA DNA complementary base pairs. And so we want to degrade our mRNA. So we'll use an enzyme that will only chop up the mRNA. And after that, we will put in DNA polymerase, which will create the complementary strand of DNA. And this creates something called cDNA, which is complementary DNA. And that's just DNA with only exons. So here are the steps that we just talked about. And you can pause here and write these down in your notes. But like we said, we allow transcription to happen, RNA splicing, we collect the mRNA and then use reverse transcriptase so that we can create DNA from it. And then we break down that mRNA and then we allow DNA polymerase to create the second strand of DNA. And of course, this creates the complementary DNA, cDNA, which is what we want if we want to create proteins from eukaryotic cells. Okay, so what if we want to identify a specific gene in the genomic library? We mentioned this, but how do we actually know, let's say we have a gene in mind and we want to know in which DNA colony it's in, which one of these plasmids it's in. So the way we do that is if we know the specific sequence of our desired gene, then we can create a radioactive complementary sequence. So we know we're looking for gene like GTAC or something. Then we would create a radioactive sequence that's complementary to that, so C, whatever it is. And this is called a nucleic acid probe. And so it's going to base pair with whatever we want. And so what you do is you take all of your genomic library and you separate the DNA strands so that now these DNA strands are kind of like single strands. And you add your probe, and wherever your probe attaches to, and that colony is going to be radioactive, so it'll light up, that's the colony with your gene that you're looking for. And so this is just a picture where we have all of our genomic library of cells, and then we have radioactive DNA probes, and we'll just pour the probes over all these colonies, and then we allow the colonies to have their DNA separate and then base pair with the probes, and then whichever colonies light up, we know are the ones that have bacteria containing the plasmids of our specific gene in mind. All right, thank you so much for watching, and we're going to continue talking about how we can use these different techniques to do pretty amazing things in lab. All right, thank you so much. Bye.